Hello, I'm Matt Sharp, AIDS cure research activist from San Francisco. I'm here today with two researchers who hardly need an introduction. They're both really integral to AIDS cure research today, and both are principal investigators to two of the NIH-funded Marty Delaney Collaboratory towards an HIV cure grant. David Margolis is a professor of medicine at University of North Carolina School of Medicine, and he's leading the CARE Collaboratory of AIDS Researchers for Eradication, one of the funded Marty Delaney programs. And from the other side of the country is Steve Deeks, professor of medicine at UCSF, Division of HIV/AIDS, and he is leading the DARE Collaboratory, Delaney AIDS Research Enterprise. Both of these projects are really the first to sort of use a collaboratory approach towards finding a cure for HIV. So welcome both of you, and thanks for your time in the middle of a busy CROI. So it's been over 5 years since Timothy Brown was cured of AIDS. Can you help us understand where we are now in the field since the news of Timothy Brown came out, and discuss some of the findings that came out of CROI that relate to that? Either one of you.
Well, I think over the last year, I think we've found ourselves at the beginning of a long road, and I think it's encouraging to see at this meeting the huge explosion in the number of people and number of approaches and amount of work being done to address, obviously this very important but very difficult problem. And there were a number of presentations, already that I think were pretty important and informative and will influence the field. I guess the one that I would start with was work from Janet Silicano and Bob Silicano's lab, a large collaboration with a lot of different groups, including many important samples from Steve, that basically tried to measure different assays that measure persistent HIV infection, so-called the reservoir, at different levels in different tests in different ways, using all the different available assays from different sorts of labs. And I think the important, very important point that study illustrated is that the problem is both smaller and bigger than we appreciated at the same time. It's the most easy to measure HIV DNA in cells, but that's actually the least reflective of the true reservoir of persistent infection. It's most accurate to measure virus that you can recover from cells. That's the smallest reservoir, but the most difficult to follow. And in the middle, it's now been shown that there are different levels of HIV RNA. That's the RNA that's not the RNA that's in the virus, but the RNA that's expressed from the integrated HIV DNA genome. And that RNA gives you a sort of middle-of-the-road picture. It's easier to detect, and there's more of it than there is of the virus that's replication competent. There's less of it, and it's harder to detect than the DNA. And some of that RNA is actually so-called probably dangerous RNA that actually will make virus, but a lot of it isn't. And so in some ways it makes things easier to measure, but in other ways it makes studying the actual problem harder.
And so do you think that after this finding that maybe researchers will go the middle of the road and use that assay more?
I think we have to spend some time figuring out exactly what we learn from each assay. I think we can do different studies and measure different things, but we have to be very precise about exactly what we've found out, depending on what thing we're actually measuring.
So we're maybe an inch forward.
Probably more than an inch, but maybe not much more than an inch.
All right. Steve, what would you find that was an advance that came out of CROI?
I think, from my perspective, some of the most interesting stuff pertaining to cure research pertain to better characterization of the so-called reservoir, where the virus is actually residing, and a fair amount of discussion, across the entire meeting about the role that central memory T cells have in in maintaining the reservoir during long-term persistence. Stuff that we more or less knew about in the past, but it really was well defined here. I mean, other highlights from the meeting, including, as David said, some more discussion about the assays. There was some exciting new data from some clinical trials, some early phase 1 type studies. And of course, there was the well-publicized case of the potentially cured HIV baby, which generated a lot of attention in the press, and I actually think a lot of healthy debate about mechanisms in the hallways and so forth. So I think, in general, from my perspective, cure research is ramping up. The amount of novel advances that are presented here are dramatic compared to what's been seen in the past. A lot of the more exciting stuff are late breakers, suggesting that a lot of the stuff is coming at the last minute. And so I just get a sense that with all this enthusiasm and excitement and all the resources now going to cure research, things are just going to take off, and we're just at the beginning now.
So what's the furthest along clinically that was reported at CROI this year in terms of cure research?
In terms of
clinical research.
Clinical research. Well, I'll comment on it because it's mainly a follow-up to David's earlier study, but probably the most advanced concept in terms of clearing the reservoir is this concept of activating the virus and pushing it out of its hiding place. The main barrier, not the only one, but probably the main barrier to a cure is that the nucleic acid, the DNA for HIV, persists in these long-lived resting memory T cells, and the current drugs don't get to it. And the only way we're going to be able to get rid of that virus is to push it out. And David's group has been working for years on this hypothesis of using a drug, an HDAC inhibitor, vorinostat, SAHA, to push it out, and showed some data that he summarized, that he presented, published in Nature recently, suggesting that you can do this with a single dose of a drug.
Right.
And, and that was the first study, and we in medicine always like to see things confirmed, and so the major advance here is work by Sharon Lewin's group, which followed up on that study and basically confirmed more or less, I think, what David found, that when you add this drug to a stable regimen, you can push the virus out of its hiding place. One of the advances that we're reporting in the meeting here, of course, is the fact that Sharon's study was able to do it over a couple weeks, and so it appears that the effect, from what I read the data, that when you do this, it's a bit sustained, and I think that was exciting. The data also suggests that the approach is safe. And I think, importantly, the data also showed that you can do this without causing, general broad activation of the immune system.
Okay. And we need to break here, right? Okay. So welcome back. We are excited to be joined now by Dr. Sharon Lewin, who is also a part of the DARE Collaboratory, but working even further west in Australia. And she's here to add to the information and discussion we just had about the vorinostat study. Welcome, Sharon. Do you want to add anything to what Steve said?
No, I thought that was really nicely a nice summary of our work. We were very encouraged, of course, to see an increase in that the virus had turned on in 90% of the patients that participated in the study. I think one interesting new finding was that we still found the virus turned on after the drug had finished, and we need to understand that a bit better. Mm-hmm. And when we looked at the number of infected cells, just using a very crude marker, we didn't see any change. Though I actually had the primary goal of the study was to see it was safe and that it woke virus up. I wasn't expecting to see a change, but we actually, over 3 months, didn't see a change in the number of infected cells. But I think we're learning that we've known for a long time that that's a crude marker of how many cells persist. And I think the work from Bob Silicano and some of the work that Steve's done with comparing different assays is telling us that we do have to look at infectious virus if we want to know we've made a dent on the reservoir.
And I have to say, I saw your presentation yesterday in the Opportunities and Threats session, and what I'm getting a lot of, or what I'm hearing a lot about and hoping that there's more excitement about news and research in inflammation. And you spoke a lot about that. I wondered if all three of you might want to talk about how that fits into the cure research model, and where we're going with that, and if the researchers and the community is actually buying into that research and interested and excited about it? How does that fit?
Well, I believe, and many people believe, that inflammation probably plays a key role in the whole reservoir story. It's, at the moment, we know that more inflammation means we can find more virus in people on treatment. And whenever you find what is called an association, you never know, is the inflammation driving the virus or is the virus driving the inflammation? But I think some interesting studies, and one that was presented here from Steve's group is telling us that perhaps in a subset of patients, the virus might be driving the inflammation. Yeah. And that's a subset of patients that we think, and I don't think this is everyone, I do think it's a subset of patients that have a little bit of virus ongoing replication on treatment. And we know that there is a little bit of ongoing replication from some of the intensification studies that are coming out. One that was published several years ago from the Spanish group, with raltegravir, and it confirmed by the group from UCSF. And both of those studies showed that after they added raltegravir, they got a reduction in activation markers.
I guess we can have this debate. I'm a little outnumbered at this table, but I'm still not really convinced that we have a definite answer in that debate. And I think, for instance, one could even cite the case of the baby as an argument against ongoing replication. So the baby is this child that was born of an untreated infected mother, treated within 30 hours of birth. Viral RNA was measured in the baby's plasma over time for the first month of life while the baby was treated during the first month of life. And then later, therapy was stopped, unfortunately, by the mother, but surprisingly, virus didn't rebound in this. Baby appears to be either a actual cure or a functional cure, and I guess we'll have to figure that out some more. I think the importance of that finding is really that it shows that post-exposure prophylaxis maybe works, and this is maybe something we need to learn about more. But I think in the regards to this particular point, that of the ongoing replication, I think it's hard for me to understand why, if there is true ongoing replication during that first month of the child's life, when there is therapy on board, but decreasing viral load on ongoing therapy, how there could be ongoing replication and then the baby be cured when therapy is stopped. If there is a source of currently infected, productively infected cells making virus with full rounds of replication, how that wouldn't persist past the time of interruption of therapy and why the baby wouldn't have rebounded. So I think this is an ongoing debate that we have to learn more about.
Yeah. I mean, I think I still believe that the major barrier is latency. No doubt about that, and that eliminating the latently infected cells is a big issue. But teasing out whether there is some ongoing replication together with latency,
still requires more.
Yeah, still is required, and the interesting evidence from the intensification studies are that if you intensify or add more antivirals, you don't change that persistent burden. So intensification will not eliminate latency, but it has taught us that in some patients there probably is some residual viral replication. But David doesn't agree with me.
It's an ongoing debate, and I'm sure as the next CROI and maybe future CROIs come forward, we'll have more information about that. I wanted to just briefly touch on gene therapy. Now, I know that you all are not actively involved in your collaboratories with gene therapy, but there was a session this afternoon or this morning that sort of laid the background for gene therapy approaches in HIV, and it was, in my mind, it was exciting because I was a part of a gene therapy trial. And I also see the implications for people like me who just want to get their T cells boosted. But my question is, do you think that gene therapy approaches are going to be used in conjunction with eradication, activation drugs, or immune modulation trials? Where is that, and how are we going to push this research and the regulators to accept that approach, because what I'm hearing is that the best approach towards a cure will be a combination approach. So who would like to tackle that?
Well, I can say something briefly about that, and then, like, you know, hear what you guys think. I think that that's, I mean, we can talk about gene therapy in specific, but I think the whole regulatory environment about how to develop and test these new approaches, particularly as they will likely be sort of combination novel approaches, small molecules, what's called small molecules, what we think about as drugs, biologics, things we think about like cytokines or antibodies, and gene therapies, cellular-based therapies. They're actually regulated in different parts of the FDA that don't really talk to each other very well, and have different levels of how they view the playing field. And that, I think, is going to make it quite challenging to do the things we really need to do to combine approaches and move forward in clinical studies.
And I hope we have word from the Forum for Collaborative HIV Research that a forum is being developed with regulators, specifically FDA, to discuss some of these very issues. Mm-hmm. So I hope that will move forward.
No, but to be a bit more optimistic here, because I totally was with David for that the gene therapy approach was going to face major regulatory hurdles, because for the most part you're going to be doing gene therapy in people on long-term therapy whose virus has been suppressed forever. And by definition, those are people who have access to drugs and can take drugs. And those people do well generally. And gene therapy is not benign. So there's this tension. The FDA and the various regulators actually have allowed a study, which I did not think was going to happen, but it is now happening in San Francisco, not with our group. Calamine, yeah, in which going to take healthy people, I mean HIV infected, untreated, but no leukemia, no lymphoma, give a little chemotherapy to make space, and do a stem cell transplant with stem cells from the study participant that have been gene modified.
Yeah.
I mean, so this is the big deal, right? And so I think they're going to be doing, about 10 or 15 or so of these individuals over the next couple of years, and so things are moving forward.
Yes.
And there is general support for doing this, and everyone's excited, and there are resources being poured into this, particularly in the state of California. But I would say that from a broader perspective, cure research. The main unmet need with cure research is the inability to deliver antiretroviral drugs to the global population for decades and decades. In order to overcome that major unmet need, gene therapy is probably not going to be sufficient. And that's why there's more interest these days, I think, in combination approaches involving small molecules or drugs that can be basically delivered anywhere.
But wouldn't you say in the short term maybe that's true, but in the long term we don't know how the technology is going to progress in terms of gene therapy?
Right.
I mean, that's my hope.
That's always...
The thing is, I think people have been saying that about gene therapy for a long... I'm ever the optimist, and the science is fascinating. In fact, the scientific session after the plenary was amazing this morning. Did you go to that?
Yeah.
With Sangamo developing, I mean, it was new to me, these peptides that link on to CCR5 and CXCR4. So the chemokine receptor still responds to the chemokine, so it can move around the body normally, but it just blocks fusion. And it was really, that was really interesting. And then another study also done in monkeys showing that you got this boost in SIV-specific T cells from the gene therapy as you got expansion of the modified cells. So gene therapy might have the science I think is amazing, but if you read the literature, they've been saying in the literature for years around gene therapy just for cancer in 10 years. It sounds like us. But, I think...
Right. And I think this is just essentially, sort of scientifically speaking, in scientific policy, this is a problem of scarce resources. And we had this in the vaccine field where for a while we were going to make an HIV vaccine with antibodies, and then it was CTLs, and now it's antibodies again. And we don't know what the future will bring, and it's hard to decide how much to invest in what sort of approaches, particularly when resources are scarce.
Well, and that brings me to my last question, I guess, which is about how the sequester is affecting your specific projects,
just
right?
cutting my budget just this morning.
So tell us a little bit about that.
I mean, we're all just getting ready to take 10% off the top. And I think this will have huge ripple effects in, you know, ability to do new things, ability to get new things funded, ability to keep things going, you know, at the current speed that they're going, having people become discouraged and moving into the field or training.
A lot of implications.
It's a lot
Do you think there's funders to fill that gap out there?
No. I mean, there's going to be some help.
Right.
I mean, my biggest concern, and this is probably just a broad issue for science, is that you know, it was a well-known story that most people who get a Nobel Prize do their work in their 30s, right? Before they're 40, younger than us. And and those people, you know, those people who are making a transition from their training to establishing their career, they're the ones whose funding right now is very tenuous, right? And they're the ones who are going to be the first to go. And so, you know, it's everyone believes that for us to advance the cure agenda, we're going to need fresh ideas from people who think outside the box, basically young people coming in with new ideas. And I know one very promising investigator who was struggling and basically had just made it, got enough funding to start up his own group, and those funds are now on hold because of the sequester, and I'm not sure what's going to happen. And this obviously has a big effect on these individual lives, but it really could have a big effect on science because it's the young people that drive stuff.
Right.
And they're the ones that are most susceptible to what's going on now. And it's going to just hurt society in general.
Right, on the whole.
On the whole.
Well, I hate to leave this on a sour note, but it sounds like there's some advances after this meeting, and it's an honor to be here with you all today and to speak with you. Thank you so much.
Thanks, Matt.
Thanks, Sharon.